Osmosis is water passing through a semi-permeable membrane. When we placed the red blood cells in the distilled water, they began to lyse because the molecules in the solution began to rush inside of the cell, which caused the cell to burst because the pressure on the inside of the cell was greater than on the outside of the cell. In the too little too late case, the registered nurse in the case put distilled water instead of the 0.9% saline solution, which caused the patient to, his cells to lyse, which is burst, because the distilled water, what is it?
The distilled water is a hypotonic solution versus saline solution, which is an isotonic solution. So when that entered the patient's veins and it was going through his blood and all that, what happened was as the water crossed the semi-permeable membrane, the cells began to lyse, eventually causing all his blood to be excreted through his urine because of the process of his organs identifying it as water and all that, causing his eventual death.
So, in conclusion, we came to the agreement that the 3% NaCl glucose and 0.9% glucose would not work because glucose is too large of a molecule to fit through the process of osmosis, so it would basically imbalance your homeostasis.
Which is why we again reverted back to the 0.9% NaCl being the IV solution that we would use for a patient, since it does facilitate diffusion and it would be able to get fluids in and out of the cell easily, whereas like we previously said, glucose is a much larger molecule and penetrating the semi-permeable membrane would be a lot harder.
Again, the effect of osmosis on red blood cells is that it allows water and materials to pass through the semi-permeable membrane as molecules go from an area of high concentration to low concentration.
La difusión simple necesita ayuda de una canal. Oh yeah, by the way, simple diffusion needs a simple canal.
What?
Help of a canal!
Oh, an ion canal.
What is it? Okay, what do I say?
Because the cell does have a semi-permeable membrane, this is what is able to filtrate what goes in and what goes outside of the cell. It regulates what molecules will come in and what molecules come out and allows our cell to maintain homeostasis throughout any actions that it may or may not have to fulfill.
When we looked under the microscope to see the blood mixed with a 5% saline solution, we saw that the cells were shriveled up. This means that the saline solution was hypertonic.
If a patient lost 1 liter of blood, in order to be able to restore the blood, we would use a total of 1 liter of 0.9% saline solution. We would use the saline solution because its osmolarity is isoosmotic and the tenacity is isotonic. Even though 5% dextrose is also an isoosmotic solution, it has a hypotonic tenacity, making it unfit for our patient.